Now that we've talked about transcription, which as you know is the mechanism by which a genetic sequence in the genome, a gene, is copied to produce a single-stranded RNA, and we discussed this in the context of producing an mRNA, which as you know is a protein coding RNA. We're ready to move on and look at the next step in the process of expressing genetic information, which will be translation of that mRNA to produce the correct protein or polypeptide that's specified by the gene. Now, we talked about the mechanism of transcription, and it's pretty straightforward. It's essentially exactly what happens when DNA is replicated. We have complementary base pairing that yields a sequence in the mRNA that is the base paired complement of the sequence in the DNA. So there's a one-to-one correspondence of the nucleotides. If there's an A in the DNA, there's going to be a U in the RNA, and so on, right? But now what we're doing is we're actually switching chemical languages. We're switching from the language of nucleic acid to the language of amino acids and polypeptides, so nucleotides to amino acids. And there's not the same one-to-one correspondence between a nucleotide in an mRNA and a given amino acid that's going to be used to build a polypeptide. The process is a bit more complex than that. And so what we're going to see happening is that we're going to need an intermediary here, an interpreter, if you will, to get us from the language of nucleotides to the language of polypeptides and amino acids, and that interpreter or intermediary in this process is going to be transfer RNA or tRNA, which serves to bridge the gap between those two chemical languages, and as we'll see, will actually deliver the correct amino acid during the process of translation. So immediately when folks started thinking about how decoding of this genetic code would happen, it became apparent very quickly that it could not be the case that one nucleotide in an mRNA would specify one amino acid. If that were the way that it worked, then we would only be able to incorporate 4 different amino acids into a protein. That makes sense, right? But as we've talked about before, we actually use 20 amino acids to synthesize polypeptides. And so using a 1-letter code in the mRNA or the gene would not be sufficient. Even using a 2-letter code, so 2 adjacent nucleotides specifying an amino acid, would not be enough, because if you think about it, we have 4 possibilities at each position, and so even using a 2-letter code would only give us 16 possible combinations, not enough to yield a sequence to specify 20 amino acids. So the minimum sequence that could be used, and what is in fact actually used, is a triplet of nucleotides, where we now have 64 different possible combinations of nucleotides, more than enough to specify 20 different amino acids in proteins, right? So that is in fact what's used. 3 nucleotides in sequence specify a particular amino acid in the polypeptide, and that 3 nucleotide sequence we refer to as a codon. So an mRNA contains codons. Each codon specifies 1 amino acid, right? And so that actually leads us into the first of several fundamental properties of the genetic code. So the genetic code, if we're thinking about how it works, we can describe some fundamental properties that define the way that it's used to decode genetic information. And that first property, as we just talked about, is that it's a triplet code. 3 nucleotides, not 1 or 2, are used to specify an amino acid. And if we think about all the different ways that we could combine the 4 nucleotides in RNA, A, U, G, and C, we get a table that looks like this with all 64 possible combinations. So this is the genetic code table, the sequence that we would see in an mRNA, and the corresponding amino acid that's associated with that 3-letter sequence, right, that codon. And immediately upon looking at this, we can see another couple of properties that really shine through about the genetic code. The first of which is that if we look at our amino acids here that are specified by these codons, we can see that for most amino acids, nearly all amino acids in fact, more than one codon, more than one sequence could specify that amino acid. And so that property is referred to as either redundancy, so the genetic code is redundant. Sometimes it's also referred to as being degenerate. So there's not one specific sequence for each amino acid. Most amino acids are encoded by multiple sequences, right? So in order to use this table, to understand the way that it works, we're looking for a 3 nucleotide sequence. The first nucleotide in the codon we read down the left-hand side of the table, the second nucleotide in the codon we read across the top of the table, and the third nucleotide in the codon we read down the right side. So for example, if we were looking for the amino acid that's specified by the codon CAG, we would find C as the first nucleotide, and that would bring us to this row. And then we're looking for A as the second nucleotide, so now we're in this box, and then we just look for the sequence CAG, and that specifies the amino acid glutamine. Right? So if we look at this and we look at the redundancy, we can see that there are actually only 2 amino acids that don't have redundancy. The first is tryptophan up here, which is specified by UGG codon, and only UGG, and the second is methionine, which is specified by the AUG codon. Right, so there's redundancy built in here. But the other converse is not true, right? The converse of redundancy would be ambiguity, and that is definitely not the case. The code is absolutely unambiguous. For any given codon, there's no question as to what amino acid is specified. The codon CGU is always going to specify arginine. The codon AAC is always going to specify asparagine. And if you think about it, that makes sense, right? It wouldn't work if when this mRNA is being decoded in the ribosome, a specific codon didn't specify a particular amino acid, then there would be confusion as to what protein is supposed to be synthesized, and that can't be the case, right? Because as we know, the sequence of amino acids in the protein is very closely related to the structure of the protein, and the structure of the protein is what determines the function. So ambiguity would not be tolerated, and the genetic code is absolutely unambiguous. The final property that you should be aware of doesn't maybe leap to mind immediately, but it's actually a property that we've already encountered in the lab in this course, and that's the fact that the genetic code is universal or nearly so. So I'm going to put universal in quotes here. And by that I mean that all organisms on planet Earth use essentially exactly the same genetic code with only a few exceptions, right? And so you can take a gene sequence from one organism and put it into another organism, and that second organism will have absolutely no problem at all decoding that gene sequence and expressing the correct protein. And we saw this in lab when we did the genetic transformation lab, and we actually took a gene from jellyfish, that green fluorescent protein gene, and put it into a completely different organism, E. coli, and saw that E. coli had no problem expressing that fluorescent protein gene. Why? Because E. coli uses the same basic genetic code as jellyfish, as human, as Discosoma, coral, and so on. Okay, so if we look at this genetic code table, we have 64 possible codons, but we see that several are highlighted here, right? So the first one that's highlighted here in green is referred to as the start codon, and it is the codon that is present at the beginning of a protein coding sequence in the gene, and it's actually where translation begins, where the ribosome begins synthesizing the protein. So the start codon always is going to be associated with the amino acid methionine, and so every single protein that's synthesized has a methionine as its first amino acid. These 3 codons that are highlighted in red up here are stop codons. So these specify the termination of protein synthesis. They're found at the end of the protein coding sequence, and they signal to the ribosome that that's the end of the sequence to be decoded, and translation ends at that position. Now, why do we need start codons and stop codons? It seems like you could just maybe begin at the beginning of the mRNA and end at the end of the mRNA. Well, it doesn't work that way for the following reason. The protein coding region of an mRNA is some internal segment of the complete mRNA. It's not the entire mRNA, in other words. So if we look at a mature, fully processed mRNA, one that has had its 5' cap added on, one that has had its poly A tail added on, and has been correctly spliced in the spliceosome, what we would see is that some region of that mRNA contains protein coding sequence, but not the entirety of the mRNA. So in fact, there are sequences to the 5' or in the 5' direction upstream of that protein coding sequence that don't code for amino acids, and that's referred to as an untranslated region. So there's 5' untranslated region, and downstream towards the 3' end of the mRNA, there's additional sequence that doesn't encode amino acids as well. So we have both a 5' untranslated region and a 3' untranslated region, and these are not read by the ribosome to build the protein. Only this protein coding segment of the RNA is used. So we have to have some sort of a signal to the ribosome that these are the boundaries of the protein coding region, and the start codon and stop codon serve as those boundaries. So if these untranslated regions don't actually encode amino acids, then what is their function? Well, we won't have time to talk about it in this class, but basically they function in regulation of the mRNA. So you could have RNA binding proteins that would recognize sequences in these untranslated regions and bind there, and in binding could do things like inhibit the initiation of translation, so slow down the rate at which this mRNA is translated, or conversely, they may bind in the 3' end of this mRNA and actually cause this RNA to be degraded more rapidly than usual from the 3' end. So they control things like translation rate, degradation rate. They can control things like localization of an mRNA within a cell, all kinds of things like that. So 5' untranslated regions and 3' untranslated regions function in regulation, but not actual protein synthesis. So we have these boundary sequences, start codons and stop codons, that signal the beginning and end of translation. Okay, so now we're ready to talk about the mechanism of translation. And as I just mentioned, we need to understand the role of our interpreter or our translator in this process. And that translator, as we just discussed, is tRNA, transfer RNA. And this is the structure of a tRNA. Like all RNAs, it's a single-stranded molecule, and as we've discussed previously in the course, these single-stranded molecules can form intramolecular hydrogen bonding interactions and fold up into these complex structures, and those structures are going to determine their specific functions. And so this is the characteristic way in which an average tRNA will fold up. Due to that intramolecular hydrogen bonding, it forms this classic, what's referred to as a clover leaf structure, right? But this is all one molecule. Here's the 5' end. We can trace it all the way around to the 3' end. And as you'll recall, these are specifically referred to as stem loop secondary structures. If we look at the structure of a tRNA in its fully folded state in 3 dimensions, it folds up into something that looks kind of like an upside down L. So you could imagine that unbase paired nucleotides in these loops, for example, might find partners to interact with in other regions of the molecule and cause this more complex tertiary structure to form in the tRNA. So there are two functionally very important regions of the tRNA that you should be aware of. And the first is this anticodon loop, which is down here at the bottom of the tRNA. And as its name might lead you to think, the anticodon is what interacts with the codon in the mRNA. So the nucleotide sequence that we find here is going to be able to match up via the base pairing rules with the codon sequence in an mRNA. So in other words, if we have an mRNA, I'm not going to write all of the sequence, that had the sequence UUC at a particular codon, this tRNA would be able to sit down and bind by virtue of hydrogen bonding interactions between its AAG anticodon and the mRNA codon UUC, right? So that's the basis of the interaction. And as we've discussed previously, this tRNA is going to be delivering to the ribosome a particular specific amino acid that's specified by this codon in the mRNA. For the codon UUC, that amino acid happens to be phenylalanine. So In this way, the tRNA sort of bridges the gap between these two chemical languages, the language of nucleotides and the language of amino acids. Right? So each tRNA has a specific amino acid that it is linked to, and during the process of translation delivers that specific amino acid into the ribosome by virtue of the ability of the tRNA to form base pairing interactions with the mRNA. Now, if we think about it, if we've got 64 codons, actually only 61 of which encode amino acids, you might think, well, we would need 61 individual different tRNAs to be able to match up with those. But in fact, the number of distinct tRNAs is fewer than that in all organisms. And the reason is that there's some flexibility and promiscuity in the base pairing interactions that can form between the anticodon and the codon. And more specifically, in this third position interaction, right? The third position in the codon interacting with the corresponding position in the anticodon loop. It turns out that there's some structural flexibility at that position, and that leads to a scenario where this nucleotide at this position can assume some different orientations, and because of that we can get some promiscuous base pairing or non-Watson-Crick standard base pairs, and these non-standard base pairs, those interactions are referred to as wobble, right? Sort of alluding to the fact that it's caused by that structural flexibility. So for example, this particular anticodon, AAG, even though we would predict that it would only be able to base pair with UUC, in actual fact, it can also base pair with the sequence UUU, because we can see a non-standard base pair interaction form between guanine and uracil nitrogenous bases because of that flexibility in alignment. And that's all we'll say about wobble, but you should be aware of it. It's an important feature of the interaction of tRNAs and mRNAs. So that's the anticodon, right? It defines the interaction with the codon in the mRNA. What about this business of being linked to an amino acid? Well, the amino acid actually gets linked up here at the 3' end of the tRNA in the region that's referred to as the 3' acceptor stem. So that would be up here. We would see a specific amino acid linked to this tRNA. Now, what's going to determine what specific amino acid is linked here is the sequence and structure of this specific tRNA molecule. And this is going to be an enzyme catalyzed reaction, right? As you might imagine. A tRNA is not just going to bump into an amino acid in the cell and become covalently linked to it. There's a class of enzymes that are responsible for catalyzing that covalent bond formation. And those enzymes are called aminoacyl tRNA synthetases, or AARS for short, aminoacyl RNA synthetase. So this is the class of enzymes that's responsible for recognizing every single one of the 20 amino acids and finding the correct matching tRNA and covalently linking the tRNA and the amino acid together, right? This is referred to as charging of an amino acid, so covalently linking it to its cognate amino acid. Now, as you can see, the enzyme, which is here in green, is going to be forming a very close interaction with the amino acid so that it's only binding to one amino acid, one specific amino acid, and then forming a very close interaction with the tRNA to sort of feel out the sequence and determine if this tRNA is the correct one that matches with that amino acid. And you might imagine that that anticodon loop would be particularly important to feel out, and you would be correct. The enzyme is only going to covalently link this amino acid to the tRNA that has the correct nucleotide sequence here. So the enzyme is going to be feeling out that sequence of nucleotides in a very high fidelity specific fashion. So what we have is a class of enzymes that recognize each of the 20 amino acids and find the correct tRNA to link those amino acids to. So in the cell, we have a pool of tRNAs that have already been linked to their correct amino acid and are ready to go and ready to participate in translation at the ribosome. So if we think about it, how many different aminoacyl tRNA synthetases do we need? Well, we need 20, one for each of the 20 amino acids that are used in protein synthesis. And that aminoacyl tRNA synthetase for each amino acid would recognize all of the tRNAs that correspond to that amino acid in cases where there's more than one. Okay, so we know the role of the mRNA, we know the role of the tRNA in this process. Let's turn our attention now to the ribosome and see how all of these components come together in the process of protein synthesis. So this is a depiction of the overall structure of an assembled ribosome. As we've talked about before, we have the small subunit and we have the large subunit in both eukaryotes and prokaryotes. This is the basic structure of a ribosome. You have to sort of imagine that these two subunits kind of sandwich an mRNA, which is this little bead-looking structure down here at the bottom, sandwich an mRNA between them, and then we see tRNA sort of entering the factory floor, delivering their specific amino acids during the process of translation. And what we see being assembled is a protein that sort of emerges out the top of the large subunit through a region of the large subunit that's referred to as the exit tunnel. Now, in terms of the structure of the ribosome, as we've talked about before, remember that a ribosome is composed of both ribosomal RNA, so a little bit more accurate structural view over here on the right, and in yellow and orange, we have ribosomal RNA, rRNA, so our second class of functional RNA molecule, non-coding RNA molecule that participates in translation, and then protein, which is performing a largely structural role in correctly orienting these ribosomal RNA molecules to interact with the tRNAs and position everything correctly. So in the ribosome, it's really the ribosomal RNA that's really functionally significant in the process of peptide bond formation and protein synthesis. And in fact, as these amino acids are linked together to build the polypeptide, it's ribosomal RNA in the large subunit that is responsible for that catalytic activity, that catalysis that leads to peptide bond formation. And as we've talked about before, when you have an RNA that functions as an enzyme, we call that a ribozyme. So the ribosomal RNA component of the large subunit has this ribozyme activity that results in new peptide bond synthesis. All right, so we're going to talk about the process of translation as it occurs in eukaryotic cells. And that process is just a bit more complex than what happens in prokaryotes, but the mechanism is very, very similar. As we talked about when we talked about mRNA processing, the 5' cap is going to be critical for initiating translation in eukaryotic cells, right? So what happens is that we have a complex assembled that consists of the small ribosomal subunit and several proteins referred to as initiation factors, which we're just sort of representing as these little blue blobs. They have their own individual names, they have their own individual functions that they perform in this initiation process, but we're just going to generalize and call them initiation factors sort of collectively. This complex of the small ribosomal subunit and these initiation factors bind to this methylated guanine cap at the 5' end of the mRNA. So we see that subunit and its little initiation factors bind here. And then what happens is something called scanning. So that complex is going to move, it's going to translocate down the mRNA in the 5' to 3' direction. And it's going to stop when it gets to the start codon, right? And that's where we're going to see initiation of translation take place. As you might imagine, the likelihood of having an AUG in an mRNA is pretty high. 4 times 4 times 4 is the probability of running into an AUG. So how does the ribosome know which AUG is the correct AUG? Well, it knows because the AUG is actually embedded within a larger sequence of nucleotides that is referred to as the Kozak sequence, after Christine Kozak, who's the person who described this sequence, right? So we can see here's our AUG start codon, but it's actually embedded within a larger sequence that signals to the ribosome, okay, this is where you need to stop, right? Stop scanning and start translating, okay? So the Kozak sequence correctly positions the small ribosomal subunit and initiation factors at the correct AUG to begin the process of translation. Now, what happens after we find that start codon? Well, we're going to see the first tRNA come and sit down and bind to that start codon. And obviously, if we have the sequence AUG in the codon, the matching sequence in the tRNA that binds is going to be UAC. It's going to come and sit down by those base pairing rules. And as we talked about before, that tRNA would previously already have been linked to a methionine amino acid by an aminoacyl tRNA synthetase, an AARS enzyme. That would have already happened previously. So this is referred to as the initiator tRNA. It brings the first amino acid into the complex. It sits down and binds with the small ribosomal subunit. And then what we see is the large subunit being assembled to form the overall complex. Now, when this complex forms, essentially what we have are 3 specific tRNA binding sites within the ribosome complex. And these are referred to as the E site, the P site, and the A site. All right. A stands for aminoacyl, and with the exception of the first tRNA, it's where each tRNA bringing the next amino acid to be added to the chain is going to come and sit down and bind. The only exception being this initiator tRNA, which sits down at the P site right next door. P stands for peptidyl. And in general, during the process of translation, the tRNA that's sitting here in the peptidyl site, the P site, is what's going to be holding on to or covalently linked to the polypeptide that's being assembled. E stands for exit. And this is the site that each tRNA is going to transit through on its way out of the ribosome. So once this tRNA transfers its amino acid or polypeptide to the next tRNA in the chain, then what you're going to see is it's going to be translocating here and then booted out of the complex, where it's going to then be out in the cytoplasm. It's going to interact with another AARC and get covalently linked to its correct amino acid once again, so it can participate in another round of translation. All right, so now that we've assembled the complex, we're actually ready to elongate this polypeptide. We have the first amino acid in the chain, so this would be the N terminus of our protein that's being assembled, and the next tRNA carrying the next amino acid to be added to this polypeptide is going to come and sit down right next door. It's going to have the matching anticodon sequence to this codon that is positioned in the A site, and it's going to be holding whatever amino acid it's supposed to be carrying that it's already been linked to. All right, and what we're going to see is the ribosome translocating exactly 3 nucleotides at a time down this mRNA, so that the tRNAs are going to be transiting through the A site, then the P site, then the E site, and then out. So here's our tRNA. It's holding the polypeptide chain that's being assembled. Here comes our next tRNA that's bringing the next amino acid in the chain. It is only going to bind if it has the matching anticodon sequence to the codon positioned here in the A site. And when it does bind, what we're going to see is a new covalent bond formed between the amino acids, right? The amino acid that this tRNA in the P site's holding is going to be linked to the amino acid that has just been brought into the ribosome. When that happens, the covalent linkage between the polypeptide and this tRNA in the P site is broken, right? So we build a new covalent bond. Here's our peptide bond being formed. And at the same time, the bond between that amino acid and the tRNA that was holding it is broken. So effectively, this entire polypeptide chain is now transferred to this tRNA. This tRNA is holding the polypeptide. Now, when the ribosome shifts towards the 3' end of the mRNA, what we're going to see is that that's going to push this now empty tRNA in that direction, and this tRNA holding the polypeptide is going to take its place, right? So we can see that happening here. The entire complex translocates down, and now it's this tRNA positioned in the P site, and we're ready for the next tRNA to come and sit down in the A site. All right. Now, this process of delivering the correct tRNA into the A site, the one that matches the codon sequence that's positioned in the A site, as well as the translocation of the ribosome down the mRNA in the 3' direction, both of those events are catalyzed by proteins called elongation factors. And just like we saw with our initiation factors, there are specific proteins, specific elongation factors that catalyze this step, sort of chaperoning the tRNA to the A site, as well as this step, translocating the ribosome down the mRNA. But we're just going to collectively refer to them as elongation factors. So elongation factors deliver the tRNAs and translocate the mRNA through the ribosome complex. Both of those steps are energy requiring. So a GTP, in very analogous fashion to what we talked about with ATP, gets hydrolyzed at each of those steps. So as you can see, protein synthesis is a very energy intensive process, right? Okay, so the ribosome is translocating down the mRNA, and at some point it's going to reach that boundary at the end of the protein coding sequence that we already talked about, that stop codon. So what happens at the stop codon, and how does that yield termination of translation? Well, when the ribosome translocates such that there's now a stop codon positioned in this A site, so in eukaryotes the stop codons would be UAG, UAA, or UGA, then there's no corresponding tRNA that binds to the stop codon. So there are no tRNAs that have matching anticodon sequences to these stop codon sequences. Instead, what binds is a protein, actually a set of proteins, called release factors, right? So this is protein, not tRNA. It's a bit of a molecular mimic. It has a similar shape to the tRNA, so it can sit down here in the A site. And what happens when the release factor binds in the A site is that the bond between the tRNA and the polypeptide that's been synthesized gets cleaved, so that covalent bond gets broken, which releases the protein from the ribosome complex, and the subunits are stimulated to disassemble from the mRNA, and the mRNA is released. This is also an energy requiring process, so termination of translation, like initiation and elongation, requires the cell to expend energy. But once this disassembly has happened, obviously translation has been terminated. All right, we're going to watch an animated view of this process that shows the structure of the ribosome and the structures of the tRNA molecules in with a bit more accuracy. But before we do that, I just want to go through a quick example, sort of tying all these three processes, initiation, elongation, and termination, together using an example sequence to illustrate how the process would take place. So here's our sequence. Let's say that this is our mRNA from the 5' to the 3' end. Obviously, this is much shorter than a real mRNA would be. Real mRNA is going to be 1,000 or more nucleotides long on average. So as we talked about before, we're going to see the ribosome and initiation factors binding to the 5' cap at the 5' end, and we're going to see scanning. So initially that ribosome, a small ribosomal subunit, would bind here with initiation factors and then scan in the 5' to 3' direction, and here's our AUG start codon, which is going to be positioned in the middle or P site of the small ribosomal subunit. So we could imagine here's our small ribosomal subunit positioned here. We're going to see our initiator tRNA come in and bind, which we know is going to be holding the amino acid methionine. And the reason that tRNA is going to come and sit down at this position is because it has the matching anticodon sequence UAC to AUG. Assembly of the large ribosomal subunit is going to occur, and now we're ready to go. So we have this tRNA with its methionine positioned in the P site, the middle site in the ribosome. A site has been opened up, right? And what tRNA is going to come and sit down here in the A site? Well, it's going to be the tRNA that has the matching anticodon that base pairs with UUU. So it's going to be the tRNA that has the sequence AAA, and that tRNA would previously have been linked already to the amino acid phenylalanine, right? So there would be a pool out here in the cytoplasm of tRNAs with the anticodon AAA already linked to phenylalanine. Once these two amino acids are positioned next to one another in the P and the A sites, we see a new covalent bond formed between them. The bond between this amino acid and the tRNA is broken, so effectively this tRNA in the A site is now holding this little dipeptide that consists of methionine and phenylalanine. This entire complex is going to be shifted down the ribosome in the 3' direction. You could also think of it as the mRNA sort of being pulled through the ribosome in that direction. And so we're going to have the small ribosomal subunit now positioned like so, right? So our large subunit is still going to be here. But now this tRNA that was in the A site and is holding the little dipeptide is now positioned in the P site, right? And we've put a new codon in the A site where we're going to see the next tRNA come and sit down and bind, and that tRNA is going to have the sequence CUG as an anticodon, and that CUG sequence encodes the amino acid aspartate. So this tRNA would previously have been linked to aspartic acid. Once again, these two amino acids are going to be linked together. The bond between this amino acid and the tRNA in the P site is going to be broken, and this whole chain now has been transferred to this tRNA sitting in the A site. Translocation, pulling that mRNA through so that the ribosome translocates down exactly 3 nucleotides at a time is going to happen. So now this tRNA with the CUG anticodon is going to be positioned here in the P site, right? That tRNA is sitting here, and it's holding the sequence of polypeptide here, aspartate linked to phenylalanine, linked to methionine, which is emerging out the top of the ribosome through that exit tunnel. And now we have yet another codon positioned in the A site. We're going to see the next amino acid delivered by the next tRNA, that next amino acid being arginine, and the tRNA that carries it would bind here because it has the anticodon sequence GCU, so it correctly matches up We make our peptide bond, break the bond between this amino acid and the tRNA and the P site, and then once again we see translocation of the ribosome towards the 3' end, and what we have positioned in the A site now is our 1, 2, 3, 1, 2, 3, 1, 2, 3 is our stop codon, right? And so CGA is our tRNA that's holding now arginine, aspartate, phenylalanine. So this is our little polypeptide that's been assembled. We now have a stop codon that has entered the A site. And as we talked about before, there's no tRNA that binds to stop codons. Instead, what we see coming in is the release factor, which stimulates the release of the polypeptide. So now our little polypeptide that's been assembled is free to leave the ribosome. Large ribosomal subunit, small ribosomal subunit disassemble, and the mRNA is released and translation is terminated, right? So that was initiation, binding of the 5' cap, followed by 5' to 3' scanning to find the start codon. We see assembly of the entire ribosomal complex, and then basically translocation down the mRNA towards the 3' end until a stop codon enters the A site, at which time translation is terminated. Okay, so now that we have run through the process of translation and sort of understand who all the players are and how they come together, I'd like to show you this video from the MRC Lab of Molecular Biology that shows an accurate structural representation of the process as it would occur in prokaryotes. But again, the mechanism would be very, very similar to what happens in eukaryotes with the exception of initiation. So here we see assembly of the small ribosomal subunit and initiation factors onto the mRNA molecule. So all of these IFs, those are initiation factors. FMET tRNA is the initiator tRNA, and here comes the large ribosomal subunit being loaded on. And you'll see the initiation factors releasing here. And what you see is that initiator tRNA positioned at that middle P site, and the next tRNA being delivered by an elongation factor. So the tRNAs in purple and magenta there, essentially being dropped off. Elongation factor releases. Now that tRNA is positioned in the A site, right next to the initiator tRNA in the P site. Peptide bond synthesis is going to take place, caused by the catalysis by the ribosomal RNA in the large subunit. Here's our second elongation factor, translocating the mRNA through the ribosome such that the ribosome has moved down the mRNA towards the 3' end, exactly 3 nucleotides, 1 codon. And here comes the next tRNA, and our first initiator tRNA, you can see exiting that E site at the other side. And here we see the tRNA sort of cycling through, being delivered by elongation factors and the ratcheting over by the second elongation factor. And here's our polypeptide chain emerging out the exit tunnel at the top of the large ribosomal subunit. Now we'd see folding of that polypeptide, into its correct structure, and then that's obviously going to lead to function. So here we see the process of termination. So a stop codon has entered, release factor comes in, there's our polypeptide being released out the top. And essentially what happens here is that a release factor comes in and binds to the stop codon, and when the elongation factor tries to ratchet over the complex, it functions sort of like a crowbar to disassemble the entire complex. So now that we have more of an understanding of how translation works, what the molecular mechanism is, how a sequence of nucleotides in a gene gets copied into the sequence in the mRNA, and how that sequence is decoded at the ribosome to yield the correct protein sequence, we can revisit this idea of mutation in a little bit more detail and see exactly how a change in nucleotide sequence can result in a change in the protein. All right, so let's take this example where we have an mRNA with this particular sequence of nucleotides, and that sequence of nucleotides is supposed to specify this sequence of amino acids in the protein: met, methionine, lysine, phenylalanine, glycine, right? It's from N terminus to C terminus. Here's our stop codon, UAA, which doesn't encode anything. So let's think about the types of substitutions, single base pair substitutions that we might have in this sequence, and what the consequence would be on the protein that's being assembled. So let's say that we have a mutation specifically in this cytosine nucleotide. So there's a mutation in the gene that results in a change so that when this mRNA is synthesized, instead of having a C at this position, you have a U at this position. What would be the consequence of that mutation? Well, if you go back to the genetic code table, what you would see is that both GGC and GGU encode the same amino acid, glycine, right? This is that redundancy property of the genetic code. So even though there's a mutation in the gene, there's actually no change in the protein sequence of the protein that's assembled from that mRNA during the process of translation, because both of those encode the same amino acid. So in that case, we'd be looking at what's referred to as a silent mutation. It has no effect on protein sequence, no effect on the amino acid sequence, and so it has no effect on protein structure or function. On the other hand, what if we didn't change this sequence at the third position, that wobble position, as we talked about before, but instead changed it at the first position of the codon? So instead of having a G at this position, we have an A at this position. Well, now if we look at the genetic code table, what we would see is that this codon, AGC, does not encode glycine. In fact, it encodes serine, which is a very different amino acid from glycine. It's one of our polar amino acids. It has a hydroxyl group as a functional group in comparison to our glycine, which has only a hydrogen atom as its R group. And so we'd be dramatically changing the amino acid. So this change in amino acid sequence is referred to as a missense mutation, where the identity of the amino acid encoded by that codon has been changed. And in this case, that would be a pretty dramatic change, changing a non-polar amino acid to a polar one. Now, what if the change occurred such that a codon that would normally encode an amino acid, say this second codon here, lysine, AAG, was changed to instead be a stop codon? Well, that would be referred to as a nonsense mutation. It doesn't encode an amino acid, it encodes a stop codon, and this would lead to what's called premature truncation of the protein. So premature termination of translation would yield a shortened or truncated protein, right? And depending on where that happened in the polypeptide chain, that could have more or less dramatic effects. But it would be premature truncation, termination of translation. So that would be just swapping out one nucleotide for another, right? But what if we now look at what would happen if we actually were inserting or deleting nucleotides in the sequence? Well, this leads to what are called frame shift mutations. Frame shift referring to a shift in the triplet codon reading frame of the protein. So remember that our start codon specifies the first codon in the mRNA that's going to be read to synthesize the protein. Well, there's no punctuation here in this mRNA. There's nothing that says, okay, this is the end of the codon and this is the beginning of the next codon. The ribosome just reads the nucleotide sequence in sets of 3 as that sequence is fed through. So this is the first codon, AUG. The very next set of 3 nucleotides, AAG, is going to be the next codon. The very next set of 3 nucleotides, UUU, is going to be the next, and so on. So what happens if we actually insert let's say, into this position, an extra nucleotide. Well, the ribosome doesn't know that that's not supposed to be there. It's going to read this triplet, AUG, as usual, and then it's going to read the next triplet, which is now UAA, a stop codon. And so this is going to lead to a nonsense mutation. It doesn't encode an amino acid. It encodes a stop codon and premature termination and truncation of the protein. So this is a frame shift. The reading frame of the mRNA has been disrupted, right? The reading frame, the frame that the ribosome reads the protein coding sequence. Alternatively, we could have insertion of, or deletion, either way, of a nucleotide that changes amino acid sequence extensively downstream from that mutation. So, for example, inserting a uracil, or in this case, sorry, deleting a uracil from this codon, which did encode phenylalanine UUU, removing that now would shift this G over to take the place of that U, right? So the reading frame once again has been shifted, and now instead of having a phenylalanine at this position, we have a leucine amino acid encoded. And next door to that, this GCU would encode an alanine, neither of which was actually encoded by the original sequence. So that would be extensive missense, so changing the amino acids downstream of that frame shift mutation. So inserting or deleting one or two nucleotides in the protein coding sequence leads to frame shift mutations, but we can actually see scenarios where an entire codon could be deleted or inserted. So in this case, we could take this entire codon that's normally present, delete that sequence through some mistake in replication or in recombination, some sort of error at the level of the DNA. And what would that do? Well, that would essentially delete that amino acid from the protein sequence, because if that sequence is not there in the mRNA, the ribosome will not make that sequence available for the tRNAs to bind to, obviously, and therefore no amino acid will be delivered at that position that should be there. And so what we have then is a missing or deleted amino acid in the sequence. In this case, we'd be missing this lysine amino acid that should be right after the methionine. Now again, what's going to be the consequence of this mutation? Well, it depends. What is that amino acid? Is it very structurally important for the protein? Is it not very structurally important? Is it at the very beginning of the sequence? If that happened here, close to the end, if we had a nonsense mutation here right after methionine, well, obviously we wouldn't get any protein at all. On the other hand, if it happened right here at the C terminus, maybe that would be a less dramatic effect that would have. So potentially it could be very, very disruptive, but it depends very much on where in the protein that mutation occurs in order to understand what the real biological effects of that change would be. Okay, so now we've talked about translation, and you understand the mechanism by which an mRNA is decoded at the ribosome, the factory floor, using tRNAs as our sort of translator, interpreter molecule, bridging the gap between the language of nucleic acid and the language of polypeptides.